This is part one of a two-part video series on DNA and RNA. We'll start with DNA. DNA is a molecule that carries the genetic instructions used in growth, development, functioning, and reproduction of all known living organisms and many viruses. DNA and RNA are nucleic acids, alongside proteins, lipids, and complex carbohydrates. They are one of the four major types of macromolecules that are essential for all known forms of life. Most DNA molecules consist of two biopolymer strands coiled around each other to form a double helix. The two DNA strands are called polynucleotides, since they are composed of simpler monomer units called nucleotides. Each nucleotide is composed of one of four nitrogen-containing nucleobases, which is cytosine, guanine, adenine, or thymine, a sugar called deoxyribose, and a phosphate group. The nucleotides are joined to one another in a chain by covalent bonds between the sugar of one nucleotide and the phosphate of the next, resulting in an alternating sugar-phosphate backbone. The nitrogenous bases of the two separate polynucleotide strands are bound together according to the base pairing rules, and that's adenine with thymine and cytosine with guanine, with hydrogen bonds to make double-stranded DNA. RNA is a polymeric molecule essential in various biological roles in coding, decoding, regulation, and expression of genes. RNA and DNA are nucleic acids and, along with lipids, proteins, and carbohydrates, constitute the four major macromolecules essential for all known forms of life. Like DNA, RNA is assembled as a chain of nucleotides, but unlike DNA, it is more often found in nature as a single strand folded onto itself, rather than a paired double strand. Cellular organisms use messenger RNA to convey genetic information, using the letters G, U, A, and C to denote the nitrogenous bases guanine, uracil, adenine, and cytosine that direct synthesis of specific proteins. Many viruses encode their genetic information using an RNA genome. Some RNA molecules play an active role within cells by catalyzing biological reactions, controlling gene expression, or sensing and communicating responses to cellular signals. One of these active processes is protein synthesis, a universal function where RNA molecules direct the assembly of proteins on ribosomes. This process uses transfer RNA molecules to deliver amino acids to the ribosome, where ribosomal RNA then links amino acids together to form proteins. DNA replication is the biological process of producing two identical replicas of DNA from one original DNA molecule. This process occurs in all living organisms and is the basis for biological inheritance. The cell possesses the distinctive property of division, which makes replication of DNA essential. DNA is made up of a double helix of two complementary strands. During replication, these strands are separated. Each strand of the original DNA molecule then serves as a template for the production of its counterpart, a process referred to as semiconservative replication. Cellular proofreading and error checking mechanisms ensure near perfect fidelity for DNA replication. In a cell, DNA replication begins at specific locations or origins of replication in the genome. Unwinding of DNA at the origin and synthesis of new strands results in replication forks growing bidirectionally from the origin. A number of proteins are associated with a replication fork to help in the initiation and continuation of DNA synthesis. Most prominently, DNA polymerase synthesizes the new strands by adding nucleotides that complement each strand. DNA replication occurs during the S stage of interphase. Then we also have transcription. Transcription is the first step of gene expression in which a particular segment of DNA is copied into RNA by the enzyme RNA polymerase. Both DNA and RNA are nucleic acids, which use base pairs of nucleotides as a complementary language. During transcription, a DNA sequence is read by an RNA polymerase, which produces a complementary anti-parallel RNA strand called a primary transcript. So transcription proceeds in the following general steps. So we have 6 steps. First, RNA polymerase, together with one or more general transcription factors, binds to promoter DNA. Second, RNA polymerase creates a transcription bubble, which separates the 2 strands of the DNA helix. This is done by breaking the hydrogen bonds between complementary DNA nucleotides. Third, RNA polymerase adds RNA nucleotides, which are complementary to the nucleotides of one DNA strand. Fourth, RNA sugar phosphate backbone forms with assistance from RNA polymerase to form an RNA strand. Fifth, hydrogen bonds of the RNA-DNA helix break, freeing the newly synthesized RNA strand. And lastly, sixth, if the cell has a nucleus, the RNA may be further processed. This may include polyadenylation, capping, and splicing. The RNA may remain in the nucleus or exit to the cytoplasm through the nuclear pore complex.